This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 124, recorded on March 11th, 2011. Hi, everybody. I'm Vincent Racaniello, and it's time for TWiV, the podcast all about viruses. Big crew today. First up, right across the desk from me, Dickson Despommier.
Hey, Vince.
How are you, Dickson?
Chipper.
Okay. Dickson's in the outer office here. He's here to stay, and now no longer any excuse for not podcasting with us.
None whatsoever.
Because he's behind me, so I can't even see him. I'll have to fix this studio, Dickson.
It's up to you, Vince.
Next up in Western Massachusetts, Alan Dove.
Good to be here.
Good to have you back. How's the West up there?
Oh, well, we're gradually losing our 3 feet of snow or whatever it was.
You still have some on the ground?
Oh, yeah.
Wow.
Yeah, and at the foot of the driveway, we still have Mount Snowplow, blocking part of the passageway.
Any rain up there?
Oh, boy, has there been rain.
Yeah, that's right.
Yeah, the ice dams took the front gutter off my house, so now the rain is particularly amusing.
Wow.
Well, moving down the coast, our next two guests are down in north central Florida, and we have Rich Condit. Hey, Rich.
Hi, fellas. How you doing?
Hey, Rich.
Hey, Rich.
We're well. You get a lot of rain down there?
A little bit. We had a front come through a couple of days ago, but no big deal. Actually, we're in a drought again, more or less.
Exactly.
Okay? But
exactly.
I'm not complaining. Everything's cool. Nice.
Well, Rich is at the University of Florida in Gainesville, and it so happens that our special guest today is also there as well, and that's Grant McFadden. Welcome back, Grant.
Hey, how you doing?
Good to have you. We had Grant on TWiV when we were in Florida. That was, I don't remember. What month was that, Rich?
I don't know. I think it was almost a year ago. March comes to mind, but that's probably wrong.
Yeah.
I don't know.
So it's Grant's second time, and we're glad to have you back. We asked
Now,
him,
is Grant in Florida right now?
Yes, I am.
Yes.
Okay.
Terrific.
I'm 2 floors above Rich Condit, so
All right.
my oxygen level is lower up here.
is that right?
We-
You're in the same building?
Yeah. We
Oh.
tried to work this out so we could do it from the same office, but the way it works out, because of the delay and the Skype, it's just too weird. So
Oh, now I thought
here
you
we
were-
are in the same building, and we just got out of a joint lab meeting together.
Cool. Yes, you're both in the same department. So Grant is a professor of microbiology and molecular genetics. In fact, you and Rich are both professors in that department, right?
That's right.
correct.
Top-heavy academics.
Very top heavy.
The poxvirus center of the universe, I tell you.
Cool.
Yeah. You had another is
Dick Moyer,
Dick
he's still
Moyer
around.
still around. It's another pox guy.
Yeah.
You're hiring more pox people?
I don't think they would let us do that.
Yeah, we never get away with it.
Well, if you hired enough of them, you could have a hockey team.
Hockey pucks?
Yes.
There you go, hockey pucks. Put that in the title,
Sad.
Alan.
It's sad.
Well, we asked Grant to join us because we'd had a request to talk about tanapox, but actually, it's just an excuse, Grant, because we're happy to have you on any time.
I'm glad to be here.
And we thought, so you suggested you'd chat a little about tanapox, and but then you really'd like to talk about myxoma.
Yep.
And if we have some time left, we'll talk about smallpox, which has been in the news lately as well.
Sure, that'd be awesome.
Before we do that, Rich has a few words.
Last week we talked about a paper from the Towers lab that noted that the integration sites from XMRV in patient samples were identical to integration sites that had been mapped in experimentally infected cell culture cells with XMRV, raising the issue that there was that in fact the patient samples represent contamination with the DNA from the cell culture. In the discussion of that, I spoke of it as if all of that work was done in the laboratory of Robert Silverman, and that was a mistake. In fact, that work represents a collaboration between at least 2 labs, most of the collaboration between 2 labs, that of Bob Silverman and Samson Chow. So the first paper where they constructed the XMRV, infectious XMRV, showed that it was infectious, and mapped the integration sites. In the patient samples, Bob Silverman is the senior author, and the first author is Beihua Dong from Silverman's lab. But Samson Chow and several people from his lab are on that paper. The subsequent two papers that we mentioned that map the integration sites both at a coarse and a fine level. In fact, the senior author is Samson Chow. The first author on the first paper is Sang-gu Kim. And he or she is also the first author on the subsequent paper. Dr. Silverman is on those two papers, and it's basically a collaboration between those two labs with substantial input for both labs. I had a conversation with Bob Silverman after that podcast went live, and he told me that they being Silverman and Chow, had become aware of this problem that the Towers Group published independently of the Towers Group. I don't know how recently, but that they were aware of it, and they were working very hard together to get to the bottom of it in the same fashion that we described, using some of the same experiments that we suggested. More or less following Vincent's recommendation to solve the whole problem, figure out all the patient samples and et cetera, and figure out what's going on. And when they've got that sorted out, they'll let us know publicly, I assume, in some sort of a publication. So I think it's our job now to let them do their work and sit back and wait for the result.
Cool. All right. Cool. Okay, well, we're waiting, and I'm sure there'll be more from other groups in the meanwhile. We're waiting for the science publication of that PATAC information that we alluded to last week.
Right.
So we can talk about it as well. But for today, let's move on to pox viruses. So tanapox, Grant, tell us all about that.
Well, tanapox is what's called a member of the Yatapox family or genus. Poxviruses are divided into 8 genera, and this is one of them. And they're sometimes called the primate poxviruses because they're only found in primates. They've only been found in humans or monkeys. And we don't know very much about them. There's 2 family members. The prototypic member is called tanapox, and it pops up every now and then in equatorial Africa. And it's a very isolated, tends to appear and disappear in people. And it doesn't cause any real serious disease. It's kind of a very mild form of smallpox that is not nearly as severe. And the other member is a virus called Yaba monkey tumor virus, which is a really cool primate virus that causes histiocytomas in monkeys, which are large proliferations of myeloid cells at the periphery. And there's some really awesome pictures of monkeys that have been infected with Yaba monkey tumor virus, in which you see these huge protuberances. And somewhere in my files, I got a picture of a monkey with one of these on its face, a rhesus macaque, and we call it our lumpy monkey slide. But it's not harmful. It tends to regress, and none of the primate poxviruses cause serious disease. They tend to appear, hang around for a couple months, and then leave. And so, consequently, they haven't been studied nearly as much as, for example, the orthopoxviruses that Rich studies, and we know much less about them. But a couple of years ago, a couple of labs, including ours, decided to investigate their biology a little bit, and we ended up sequencing a couple of them, and another group in England also sequenced one of them. And it turned out to be full of cool genes, the sort of things virologists love to ponder over and tinker with and play with. And it actually led to a couple of curious and interesting stories. And you've alluded to this previously in one of your podcasts. But let me back up and tell you about a paper that was published by a fellow named Karim Assani, who's at the University of Western Michigan in Kalamazoo. And in the mid-1990s, Karim published a really cool paper showing that tanapox-infected cells secreted something that inhibited a whole bunch of cytokines. And his first paper showed that something that tanapox made would inhibit IL-2 and IL-5 and interferon gamma, and seemed to do it all at once. And but one of the problems was nobody, including Karim, knew what the protein was. We didn't have the gene. And so consequently, the paper got published and then sort of languished for a while. And then Karim came up with a second paper showing that there was another activity in tanapox-infected cells that inhibited TNF. And at the time, Karim thought it was the same protein, but he was getting so interested, but at the same time, it was hard to make progress without the gene. So it was around then that Karim called me up and said, can you help me find my magic protein? And so we talked about it, and the way it worked out was we were in the middle of sequencing one of these viruses, and we said, well, if we can learn something about the protein, maybe then we can identify what the gene is. So Karim went back and took some extracts from tanapox-infected cells and poured it over a column that to which was affixed human TNF. And so he simply bulk purified the TNF binding protein, eluted it off, and got an N-terminal sequence. And so we took that N-terminal sequence and went looking for the gene, and lo and behold, there it was in the genome. It was a gene called 2L. And but
classic biochemistry.
That's fantastic stuff. Yeah, love it.
Bucket biochemistry. You just go seek it out.
Yeah.
But the amazing thing was when we looked at the gene, it didn't look anything like any of the known TNF inhibitors. In fact, what it looked like was a stolen MHC class 1 molecule. In the sense that it looked like sometime in history the virus had stolen this as an immune molecule and then had reconfigured it into a cytokine inhibitor. So when we got this result, we actually had to go back and change the annotation of genes for that virus. Because initially, when we had found the 2L gene, we had thought it was something involved with the inhibition of cytotoxic T cells. In other words, immune recognition, something like that. And yet, what the virus was using it for was a secreted sponge of human TNF.
This is secreted and soluble. Is that right?
Right. Now, normally, MHC class 1 molecules are stuck in the cell surface. And but the virus had taken it and grabbed what's called the alpha-1, alpha-2, and alpha-3 domain, the ectodomain, or the part that's outside of a cell, and had turned it into a secreted molecule.
Nice.
Is this specific for tanapox virus, or do other pox have it?
Only other Yatapox viruses have it. So it's pretty specific for Yaba monkey tumor virus and tanapox. There's something that looks a little like it in molluscum contagiosum virus, but it's quite a distant relative, and we don't even know if it binds TNF. But this particular molecule, it is awesome. It binds human TNF with incredibly high affinity. In fact, much higher affinity than the known commercial inhibitors of TNF, things like Enbrel or Remicade, that are used as drug treatments for TNF-driven disease. This, viral molecule beats them amazingly in terms of affinity and ability to bind and inhibit human TNF.
Wow.
Evolution beats intelligent design.
Boy, I'll tell you, viruses have been doing R&D, and they do it well.
No question.
Well, and they've got the most brutal product testing procedure.
Yeah, living and death. It's a great selection.
Has this been trialed in humans to replace Remicade, for example?
Well, I should disclose here that this particular molecule has been licensed off into a biotech company that I co-founded, a company called Viron Therapeutics. So I should just sort of say that from the outset. But yes, it's slowly being developed. It hasn't hit human clinical trials yet. It's still in what's called preclinical testing. So far, there's only one viral protein that I know about that's actually been tested in human clinical trials to treat disease, also done by the same company. It's a molecule called SERP1, and it just finished its human phase 2 clinical trial.
Because Remicade is incredibly successful, and it would be interesting to see if this replaced it. We were just talking yesterday, the developer of Remicade, or the discoverer of the antibody against TNF, Jan Vilcek, who's here in New York City.
Yeah. Now, both Remicade and Enbrel were created as specific TNF inhibitors. One is a monoclonal antibody, and the other is what's called an FC fusion of an ecto-binding domain of TNF. And they both work very well. They're both good recognizers and inhibitors of human TNF. But this viral protein will do the same thing in with 1/10 or 1/100th the amount of protein, because its affinity is so much higher for TNF than those guys are.
What would you use it for? Treating what kind of disease?
Well, right now Enbrel and Remicade are used extensively for rheumatoid arthritis, and they're making some inroads into in some of the inflammatory bowel diseases. But it's possible that there are other systemic diseases out there that are dependent upon TNF as well, that such things could sponge up the TNF and do a good job for. Clinical practice tends to work that a drug gets into the market based on one thing, and then as clinicians learn how to use it, it migrates laterally into other conditions depending on how it works in people.
Most of the pathology from severe cerebral malaria is due to the induction of TNF. So,
okay, I didn't know that.
If you could put that into that equation, you might have a very interesting way of intervening with.
So is that TNF in the brain, or is that
systemic?
It's
in the brain.
TNF?
No, it's in the brain.
So probably you'd have to get the drug into the brain as well.
That's probably the biggest. That's the drawback there. I think.
The blood-brain barrier would be your enemy there.
Yeah, you bet.
Yeah, and if you can figure out how to get a protein-based drug into the brain.
No, you're right. You're soaking it up from the circulation is a lot different than this one. You're right.
If you take this gene away from tanapox, what's the consequence?
Well, we don't really know, because we've never really gone into primate models. The only model that you really have to examine what the virus does biologically is a human or a non-human primate, and we just never went down that road.
But you would predict that the virus wants to inhibit inflammation, right? So this is an important component, so it probably wouldn't do as well in a host without it.
Well, we do know of other classes of TNF inhibitors in other viruses. For example, the virus we study, myxoma, has got a TNF inhibitor in it, but it's rabbit-specific, and it's a completely different kind of molecule. It's a mimic of the TNF receptor, but it seems to have been picked up by the virus to do the same thing. And when we knock that gene out of myxoma, that virus is clearly attenuated. It can't compete in rabbits anymore.
So going back to the beginning, tanapox and Yaba monkey tumor virus, how are they transmitted? What's their natural host reservoir?
Well, we know almost nothing about that question. We don't know what the animal reservoir is. In fact, it's never been seen in anything but humans and non-human primates. So it's a little bit of a mystery what its long-term reservoir would be. We have one little clue, and it's just a clue, that when we look at that TNF inhibitor that I just described to you, that secreted class 1 molecule that soaks up TNF, when you look at it, its ability to inhibit TNF from other species, it can only bind and inhibit monkey or human TNF. It doesn't inhibit murine TNF, for example, which sort of tells me that its reservoir maybe isn't a rodent. It may simply be another primate, and we just don't know what it is.
And so there's some evidence that Yaba monkey tumor virus is mosquito-borne. Is that correct?
Well, the lesions pop up every now and then in exposed skin in times when mosquitoes are at their highest. So it's been guessed that it's possibly biting mosquitoes that is transmitting it from host to host. That's just a guess.
That would be the first example, though, of a pox virus transmitted by mosquitoes, wouldn't it?
No, actually, myxoma virus is also transmitted very well by mosquitoes. Yep.
But is that the normal route, though?
Well, it is normal in Australia right now, where there's lots of mosquitoes and lots of rabbits. But evolutionarily, it was probably transmitted by all kinds of biting arthropod vectors.
Aren't we glad that smallpox wasn't like that?
That's right. Smallpox did not have such a vector.
From your description, it sounds like tanapox is a systemic disease. Is that correct? As opposed to some...
It possibly has some systemic component, but the lesions tend to be very singular or small numbers.
Okay.
So it's not quite like the systemic viremia associated with smallpox or things like that. It looks like it's a local lesion, and the virus persists for quite a bit of time. And if a mosquito comes along and bites, let's say, a lesion and goes on to bite another primate, that's probably how it gets transmitted.
Okay.
So what's up with tanapox now? Are you still working on it, or is this the end of the line?
There's not a lot going on with it right now. It's a really interesting virus. It's full of cool molecules. It's got a chemokine receptor embedded into its viral genome. It's got an inhibitor for IL-10. It's got an inhibitor for IL-18. It's a really cool virus, but it's hard to do biology on it unless you want to work with non-human primates. And so there's certain limitations to it as a model system, as a virus. But taking the genes out and studying them one at a time, a number of labs are doing that. But it isn't one of those medically important viruses that immediately leaps to, let's say, the attention of study sections.
Although if you get an effective TNF inhibitor out of it, then maybe it's a pretty medically important virus.
Yes, and time will tell how it actually behaves as like a drug. But the precedent with SERP1 is looking very promising. So more of these molecules may be coming down the pike.
Grant, how many genes does it have, by the way?
It has about 140 genes, something like that.
Wow.
So that's not big by poxvirus standards, but by virus standards, it's full.
And my usual guess is that roughly half of these genes are going to be involved in messing with the host. They are not, immunomodulatory genes, host range genes of one sort or another. So there's a huge investment in tinkering with the host.
Absolutely. And in fact, I think even now we still underestimate the diversity of how these molecules go after the immune system. Whenever we study one individually long enough, we almost inevitably find there are other targets and other things that it does. The old days of T antigen for SV40, when it was first discovered, it was thought to be just a simple oncogene. And then as more and more people studied it, the list of things that it would do biologically just grew and grew. And each one of these, exactly. And these modulatory proteins, they appear to be like little T antigens as well. As you study them, they've got more targets than we realize.
Do you think they might even be used to discover new cytokines that haven't been identified yet?
Entirely possible, because some of them have got targets or ligands that we don't know what they are.
Exactly.
And so they possibly could be used as fishes for new immune molecules.
Yeah, the virus has been studying immunity a lot longer than us.
Exactly. As one of Dick Moyer's graduate students put it once, through evolution, the viruses have discovered the pressure points of the immune response. And so if we have a look at what they're doing, we'll understand what the critical features of immunity to pathogens really is.
Right. And the viruses who flunked their tests are no longer with us.
That's right.
It's a jungle out there.
I tell you.
Yeah.
Wow, a lot of quotable things here today.
Indeed.
Well, maybe that's it for TANF, and we should move on to myxoma.
Sure.
I think you talked a bit about myxoma on TWiV 84 when we visited Florida. You talked about the rabbit story
Yep.
in Australia. But you didn't talk about the purging, so maybe we can get onto that one.
Yeah, one of my big passions right now is trying to understand myxoma virus. It is in nature, it's a rabbit-specific virus, and the only thing it ever infects or causes disease in are rabbits. And it's never been really clear why the virus was particular about rabbits, because it's quite common in virology. When you take these viruses and study them in a lab in tissue culture, sometimes these specificities or tropisms seem to vanish. In other words, the virus is more promiscuous in cultured cells than it is in real biology. And one of the things we have been investing ourselves with over the past couple of years is that we stumbled upon the fact that the virus grows very well in human cancer cells in addition to rabbit tissues. And so we've been looking to see whether or not we could exploit what we've learned about the virus in rabbits to infecting and killing cancer cells. And maybe I should back up a little bit and just sort of say a couple words about oncolytic virotherapy in general, because I don't know how often you've talked about it on TWiV.
No, not very much, so that would be great.
Not much.
Yeah.
Okay, so oncolytic virotherapy simply means using a live virus to infect and kill cancer cells, both in culture in the lab and also in animals or in people that have cancer. And the basis of oncolytic virotherapy is that there's something different about cancer cells that makes them more infectable by given viruses than their normal somatic cell counterparts. And the field of oncolytic virotherapy has been around for, probably a century, in the sense that people have noticed that cancer will sometimes regress following a virus infection, at least for a while. So as a result, a number of viruses have been investigated, particularly those that have a natural ability to infect cancer cells and tend to grow better in them than in normal somatic cells. And we still don't understand why that is the case, why cancer cells seem to be generically much more susceptible to viral infections than normal cells. But it may be part and parcel to how cancer cells come about, how cells become transformed. It's possible that one of the consequences of becoming a cancer cell is that it loses its capacity to engage in antiviral signaling pathways, for example. It's like a cost benefit for a cancer cell.
Well, and in fact, you see some evidence of that in most tumor cell lines that people have really looked at, right? I mean, the interferon pathway is mucked up or something else.
Yes, and I think we're just still beginning to learn the number of pathways that have the capacity to be antiviral, but that are compromised in cancer cells. But it's a generic observation that there are many viruses out there that do tend to grow better in cancer cells than their normal counterparts. And myxoma happens to be one of those. It just happens to be a rabbit virus. So we and others have been exploring different kinds of cancers, different kinds of models to see, are there cancer types that might be particularly susceptible to using viruses to eliminate cancer. And by and large, people who do this kind of thing, the viruses fall into two groups. One group are human viruses, and these are viruses that are found in people that human beings around the world have been infected by them, sometimes, subclinically. Things like adenovirus, for example, it's spread around the world. There are adenovirus mutants that have been devoted into oncolytic virotherapy. And in fact, there is a virus in China that's been licensed by the equivalent of the Chinese FDA for oncolytic virotherapy for head and neck cancers. But the other group of viruses are what we call the non-human viruses, and myxoma is one of them. And these viruses tend to be animal viruses. They don't tend to normally infect humans, but they have a natural or biologic ability to infect and kill cancer cells. And the reasons for that may be quite different from one virus to another. And so their mechanisms of action may be quite different from each other. But at the moment, people are looking for what are the best cancers, what are the best types that might be susceptible for oncolytic virotherapy. And in particular, how do you deliver the virus to the given cancer? And delivery is a big problem, or I would say the number one challenge of oncolytic virotherapy right now. How do you get the virus to all of the cancer cells that you want to get it to? Most cancers, the problem isn't the primary tumor. The problem is the metastatic sites that distribute themselves around the body. And it is a technical challenge of how you get virus to all those different sites. And so a couple years ago, we went looking for a cancer model where we could control the access of virus to all the cancer cells. And that's how we ended up studying whether or not we could use viruses for what's called bone marrow transplantation. And to make that understandable, I should probably back up and just say what bone marrow transplantation is used for. Patients who undergo standard therapy, let's say irradiation, drugs, for whatever cancer, certain cancers tend not to respond very well and relapse of the disease occurs. But for some kinds of cancers, there is a last-ditch strategy that's called myeloablative therapy. And this is particularly true for some leukemias, some lymphomas. And what it involves is if a patient has failed standard chemotherapy or the standard regime of drug treatment, and the cancer has come back and it's spreading through the body, one possibility is to give those patients what is, in essence, lethal chemotherapy, very high doses that hopefully are enough to kill all the remaining cancer cells in the patient. But the downside of that is that those very high doses also wipe out the immune system. That's why they're called myeloablative. And consequently, if that chemotherapy does work and does clear out all the cancer cells, you have to rescue the immune system of those patients with an immune system. In other words, hematopoietic stem cells needed to rebuild that patient's immune system. And you can get those stem cells either from the patient themselves that you harvest before you go into the high-dose chemotherapy, or you can get it from a related donor. So when it comes from the patient themselves, it's called autologous. In other words, autologous stem cell transplant. And when it comes from another patient, it's called allogeneic.
And it doesn't actually have to be a related donor. It can be somebody who matches on all of the...
Exactly. There has to be a close enough MHC or HLA match such that the graft well, there's a couple things that graft can do. One of the dangerous things is the graft can actually attack the patient.
Right.
And that's called graft versus host disease, and it can kill the patient. So a mismatched allogeneic graft can be a very dangerous thing, and clinicians have to be very careful about managing allogeneic grafts. Autologous grafts...
Basically, the immune cells from the graft attacking the host, the patient.
Exactly. And it can be very life-threatening. It can kill the patient. So
the one that everybody's familiar with, the boy in the bubble down in Texas, that's exactly what happened to that poor little kid.
Well, having no immune system is a disaster. And
Exactly.
It's true with HIV patients. It's true for transplant. You have to be very, very careful. The absence of immune system can be very deadly. So clinicians, if they have the option, much prefer to use autologous grafts to replace the stem cells. In other words, use the patient's own stem cells. And generally, they're obtained from the bone marrow or sometimes from what's called mobilized peripheral blood. But the point is you take the stem cells, the hematopoietic stem cells, out of the patient before the high-dose chemo, freeze it down, give them the high-dose regime, let them recover, and then you reconstitute the immune system back into the patient. So when it works, it can it can save a patient's life. In fact, my brother-in-law, Bill Browett, his life was saved by a stem cell transplant, just like that.
Wow.
He had Hodgkin's lymphoma about 10 years ago, and it saved his life. He's happy and has a great life right now. And so when it works, it can be life-saving.
Is this usually done for the lymphoid-type tumors, or it can also be done for solid tumors?
It tends to be used for leukemias and lymphomas right now. In theory, it could be used for any cancer where the chemotherapy actually could kill all of the what's called the residual disease in that patient.
Right.
I know it has been done for solid tumors because my sister-in-law had one of these for a cervical carcinoma 20 years ago. I don't know how much it's done currently.
Clinically, right now, what oncologists find is that it's most useful for leukemias and lymphomas right now. But one of the points I was trying to make is that one of the reasons that it can fail as a method is if the graft itself, the bone marrow graft, is contaminated with cancer cells from the patient. Because then what you've done is you've frozen them down very nicely,
Right,
and then
protected them,
reintroduced
put them right back in again. That's right.
And you put them back in.
Arg.
And so when I arrived at the University of Florida, I met a number of colleagues, including Chris Coggle and Ed Scott and a few others, who are hematic stem cell transplant experts. And they were telling us about this problem of cancer cell contamination in bone marrow grafts. And people have tried for years to delete cancer cells out of bone marrow grafts or stem cell grafts, and have mostly failed to do it well enough to be useful clinically. And so we were wondering, if we don't know how to do it right now technologically, maybe there's a virus out there that's smart enough to be able to do it. So that's how we ended up doing pilot studies with our myxoma virus to see if we couldn't use it to eliminate cancerous cells out of bone marrow grafts. And one of the reasons I really wanted to pursue that direction is it's one of the few cases where we can deliver the virus to all the cancer cells that we want, because we do that outside of the patient. It's called what ex vivo infection. It's taking the sample in the lab. So we can get the virus to every single cell. And we get around, or we circumvent, the delivery problem, which is one of these issues in oncolytic virotherapy. How do you get the virus to all the cancer cells that you have to get it to? So in order to be successful, the virus has to do two things. One is it has to leave the normal stem cells alone. It can't hurt them. It can't stop their ability to reboot an immune system. The other thing it has to do is it has to recognize the cancer cells and eliminate them by some fashion. So that's what we've been doing for the past couple years in my lab. And we've published one paper so far and one review, and we're in the midst of a variety of other confirmations to show whether or not this virus can selectively delete cancer cells from bone marrow or stem cell grafts. And that's what we're in the middle of right now.
So I remember when I talked to you in Florida, you had done some pilot studies in mice, right?
Correct.
Where you could take out, you could, I guess, put a tumor into mice of some sort or induce it and then take out bone marrow and then show that you can get rid of that tumor by infecting the bone marrow in vitro.
Well, actually, we've got a couple different kinds of models ongoing in the lab right now. The bone marrow projects, we use mice as sort of a surrogate recipient for how good the stem cells really are at engrafting properly into the bone marrow of a recipient. So for those experiments, the mice are sort of indicators of the potency of the stem cells, because they're the best surrogate we have before going into actual patients. And I should say, we haven't gone into people yet. This is all done using surrogate mice as the recipients for these grafts. And we use the engraftment as our readout of whether or not we've hurt normal stem cells or we've deleted the cancerous versions of those stem cells. We also use mice where we set up tumors and set up different kinds of cancers to ask whether or not we could use the virus to go in and clear those tumors out. And the models we're exploring right now in the lab actually are for disseminated pancreatic cancer. But those were the case where we set up the tumors in the animal and then ask, can we use the virus to clear those tumors?
So when you're doing the ex vivo infection of bone marrow, is it 100% effective or is there something less?
Well, right now, we're still learning how to do it. So, for example, when we do it with normal stem cells, we find that the virus is pretty harmless. It doesn't seem to hurt the normal CD34 positive stem cells that we get from normal donors. When we do the same experiment with cancer cells, and we've looked at both primary patient samples that come directly, for example, from a patient with acute myeloid leukemia, or we look at cancer cell lines that have been grown in the laboratory and are used as surrogates of the primary disease. And I should say that sometimes we find the cell lines don't act exactly the way primary patient cells act. But having said that, being able to test both kinds of cells is very, very valuable. And what we found with primary patient samples is the virus does a very good job of cleaning them out, of preventing their engraftment into recipient mice. For the cancer cell lines, we find the results depend upon the cell line. And then we get into issues of what is the virus really doing? How is it deleting these cancer cells? And this is the sort of thing we're involved with right now. We find some cell lines are really susceptible for purging and some cell lines aren't. And we're trying to learn what's the difference between them and what is the virus actually doing.
I'm always suspicious of cell lines, in this way.
You need to do both, I think, to make progress. Cell lines have the big problem is that they evolve and they change in tissue culture, and some of their properties are surrogates of what they were originally in the cancer, and some of their properties aren't. But the problem with doing everything with patient samples is once you run out of a sample from one patient, you can't repeat the experiment.
Right.
You can only go to another patient.
I suppose also from the cell lines, even if they're not perfect, you can learn something about mechanism.
Yes, and in fact, you can learn some basic fundamental principles from cell lines, but then, of course, the onus is on you to take whatever you think you've learned and go show that it's true in primary samples as well.
Sure. So, Grant, with cancer cells versus normal cells, does the virus get into both cells and then something happens after that, or does it just get into one cell only?
If you had asked me that question 5 years ago, I would have said pox viruses are promiscuous for their ability to bind and enter any mammalian cell, independent of the species. That's because pox viruses have got these really cool, it's called an entry fusion machine on their virion. And that makes them really good. If they can bind to a mammalian cell, they're very good at fusing with the membrane and dumping their contents inside of the cell. So they tend to be much less dependent upon things like receptors than other viruses that can be really particular and very specific about needing virus receptors to get into its target cells. So pox are much more promiscuous than most other viruses.
Is that because they have an envelope that they got from the host cell to begin with?
They have an envelope, and it's not. It does come partly from a host, but it's been highly modified by the virus.
Sure.
So
the answer to my original question was it gets into both cell types and then something happens in the cancer cell.
That's what he thought
doesn't.
5 years ago.
Okay, but today,
sounds like.
Fast forwarding today,
what
Yes.
I would say is that in many cases, that's the answer.
Okay.
That it binds and enters both the normal and the cancerous cells, but in the cancerous cell, the cells have got a defective antiviral signaling pathway, and the virus makes it all the way through to its replication cycle.
Got it.
Whereas primary somatic cells are much more robust in how they can block the virus from continuing on its replication cycle. But now I would say, based in fact on these studies I'm telling you about, we're starting to bump into cells, cancer cells, where the virus can't bind to them. And we're starting to suspect that normal human stem cells are the same way.
Have you ever tried to push the virus so it will replicate in a human cell?
Well, it can replicate in human cells in culture. In other words, if you take a primary human fibroblast and take it away from all other elements of the immune system, including cytokines, so in other words, you isolate that fibroblast, the virus can get in and replicate quite well, as a matter of fact. But as soon as and that's a fibroblast. If you do the same experiment with, let's say, a myeloid cell, like a human macrophage, the virus doesn't replicate because those cells turn on cytokines like interferon and TNF, and that aborts the virus replication.
What about latency? Does this virus ever become latent in normal cells?
No one has ever seen a pox virus become latent in that sense because it doesn't go to the nucleus. It doesn't integrate. It lives out in the cytoplasm. Okay. So if the virus isn't being actively aggressive about promoting itself, it tends to go away. It tends to.
So am I right then in assuming that there are no remnant pox virus sequences in human genomes?
As best as we can tell, although it looks like pox viruses have stolen some genes from hosts.
Right.
So that's in the other direction.
Right.
But we don't see endogenous pox viruses. Put it that way.
So you don't think it's a problem that you might select for viral variants that can replicate in some bone marrow cell?
Right.
Well, viruses are biology, and they're replicating entities. And they're subject to Darwinian forces like all of biology. It is an important topic in the sense that the whole field of oncolytic virotherapy, everyone who studies live viruses to infect cancer thinks about this issue. Could the virus ever evolve, for example, into a form where the tropism was different or that was radically different from the virus that we put in to begin with? So most people think of oncolytic virotherapy strategically as a short-term strategy where treatments would be for windows of time, not for months and for years, but for windows of time, very much the way drug therapy is currently used for treating cancer at the moment. But the honest answer is this is something that every virus that ever makes it into the, let's say, human condition or human trials, this is something that everyone pays close attention to. What is the status of the virus going in? What is the status of the virus coming out?
Right. I once had the privilege of just listening to Dr. Fenner talk about his experience with the rabbit problem in Australia. And
right.
I wondered if you could just summarize in general what actually did happen at the end of all of that. Did the rabbits become resistant to myxomatosis? That's the one. Or did the virus actually change? Which way did that go? And what is the status of the rabbits in Australia today? I've seen that movie about extending that rabbit fence all the way across Australia, so I guess they still have a little problem with them, don't they?
Oh, it's more than a little problem. It's still a big problem. You mentioned Frank Fenner, of course. You all know he just died a few months ago.
Yeah, exactly.
We did a little obituary to him.
Right. He was one of the great virologists of this century.
Indeed, a gentleman, too, a wonderful man.
Yes, in the original sense of the term gentleman scholar. He was a very erudite and gentle human being. He was one of my heroes.
He would have loved this show, by the way.
Absolutely. And in fact, as we were getting into using this virus to treat cancer, I had some communications back with him. And
nice.
if he'd been younger, he would have just loved this unfolding story.
No question.
But getting back to the issue of myxomatosis, that virus was released in 1951 into Australia to try and control the feral European rabbit population that had gone just crazy in Australia. And the virus ended up being transmitted from rabbit to rabbit by mosquitoes and actually made incredible penetrance into the populations in Australia, killed huge numbers of rabbits back in the 1950s. And then coevolutionary pressures took over, and it worked on both the virus and the rabbit. So there was a gradual selection for what was probably an allelic subpopulation of rabbits that were originally a little more resistant to the virus, and they became more and more predominant. And nowadays, the rabbits that are hopping around in Australia are much more resistant to the killing effects of the virus. But they're still highly infected, and they transmit the virus very, very well. But at the same time, the virus attenuated, or its virulence properties changed according to the host as it changed. And so the virus that's out in the field right now is actually a population of viruses that vary in what we call virulence, their ability to infect and kill naive European rabbits. And that coevolution is going on to this very day. It continues as we watch.
Amazing.
So what has to be done with myxoma as an oncolytic virus before it will go into people?
Well, first, a couple of things need to happen. One, of course, is you need to have virus that it's what's called GMP. It's clinical grade virus that has passed all the strictures of FDA in terms of purity and all the things that you have to do for quality control before would be allowed to go into people.
Well, and the whole manufacturing process has to be.
The whole process would have to be approved by the FDA. And this is what happens in all of oncolytic virotherapy. There are a number of viruses that are out there in the U.S. in clinical trial right now, and they've all had to go through this process of GMP manufacture, approval of the FDA, approval by the local IRBs, for example, that would oversee the trials. So you first of all have to have a clinical grade virus. The second thing is, in our case, we probably would pick a mutant of the virus that can no longer hurt rabbits. So we've got such things in the lab that are completely harmless, often because we knock strategic genes out of the virus. And although it affects the ability of the virus to cause myxomatosis, it doesn't hurt its ability to be oncolytic. A virus like that is what we would go forth to, let's say, the FDA to use as our what's called the lead candidate.
Right, because you want to use the weakest virus that can still kill the tumor cells.
Exactly. We would like a virus that can't survive in biology. In other words, can't get out and propagate in anything that we know of, and only replicates, as best we can tell, in cancerous tissue.
Right.
Do you have such a virus in hand?
Yes, we've got several candidates, and we're trying to decide which one of the several candidates looks to be the best for going forward to the FDA.
How do you make GMP virus? Do you have to have a company make it for you, or you have to partner with someone or contract it? How does that work?
The easiest way is to contract it out to people that are professional at this. It is possible, like in an academic setting, to make GMP virus, but you really have to know what you're doing. And for a new virus, for a new process where you're establishing the conditions from scratch, it's a lot easier to do it in an entity that's been doing this for years. Because as you know, viruses have been made for GMP manufacture, for gene therapy, for vaccines, for all kinds of uses. And oncolytic virotherapy just happens to be the newest kid on the block.
Yeah, and it's important to note that GMP is not just wash your hands before you go into the lab. It's an incredible pain in the tuchus to set up a GMP lab.
Absolutely. You have to document everything and to an extreme that is far beyond what we tend to do in our own research labs.
I think
we got Rich Snyder to do this here?
Yes, and in fact, I've already talked to Rich, and he's he has the ability and the facility is ready to go. So our next task is to get the money and get the process worked up, and that takes time.
Just to fill everybody else in, there's a facility here for doing this kind of thing that was set up, in fact, because there's a lot of adeno-associated virus gene therapy work that goes on at UF.
Right.
And so this facility was constructed to do that and a bunch of other stuff, and it's actually become part of an educational facility as well to train people to work in a GMP facility.
Right. Fortunately, we know a fair bit about how to make a GMP virus that is satisfactory to the FDA, because a number of oncolytic viruses have been through that process and are in trials right now. For example, there's a company in San Francisco called Generex, and I should disclose I'm on their scientific advisory board, which is why I know a fair bit about their platform. But they're using vaccinia virus as an oncolytic, and one of their target indicators is liver cancer. And one of the things about liver cancer is that if you take anything and shoot it into the blood system, the liver's job is to filter the blood system from stuff that gets into it. And so it turns out that intravenous injection is a great delivery mechanism for liver cancer.
But isn't the cancer of the parenchymal cells rather than the Kupffer cells?
Of course, it depends upon the liver cancer, but the way the filtration works is that the virus, of course, once it gets into that site, can replicate from cell to cell. So it actually can find cancer cells remarkably well.
So it sounds like you're maybe less than 5 years away from a phase 1 trial. Whenever you give those things, it ends up biting you. I would love to say within 5 years. I would love to say less. Yeah.
But the reality is there are these hurdles in front of us. And so all I can say is we're going to approach those hurdles with all the energy we have and be honorable with our presentations to the FDA, et cetera. And how long it will take is probably how long the process takes to unfold.
Right.
And so in the future, we can give you bulletins, but right now I can just say that I'm optimistic and ready to take it forward.
I'm curious about the first batch of patients. Would they be healthy patients or terminally ill patients, do you think?
Generally, FDA wants to see safety trials. In other words, usually the purpose of first trials are to show that your method, or in this case, your virus, is safe.
Right.
Because the mantra at FDA is safety, and then safety, and then safety, and then efficacy is next down the line. In our case, the way we would probably go forward is normals don't get bone marrow transplants. The people who get bone marrow transplants are those that have cancer and are queued up for bone marrow transplants because of the nature of their disease. So our first patient cohorts would probably be those that are already queued up for autologous bone marrow transplantation. And we would probably go in with a strategy in combination with FDA and the IRB that would, we think, ensure the safety of those patients, and would probably be a dose escalation from very low doses up to higher doses. That's generally how clinical trials work.
Yeah, for some cancer drugs, the way they approach this, if it's something like this, where it's just not a procedure or anything that you would give to healthy volunteers, they may authorize the phase 1 to be done in actual patients who have failed the first-line therapy.
Exactly, and quite likely that would be the scenario for us.
So compassionate use patients.
Kind of.
Kind of.
'Cause it's patients who, particularly in a case where failing the first line therapy pretty much means you're going to die.
That's right.
Then that kind of addresses a lot of the ethical issues that you would have. Well, if the thing is toxic, then they're no worse off than they were before.
It really reminds me, Vince a little while ago recommended a book as part of his recommendations called The Emperor of All Maladies.
Mm-hmm.
It's a book about cancer and about the history of cancer. And actually, I read that book about the same time you recommended it, and it's a fantastic read. And I'd recommend it to anyone. But the author, Siddhartha Mukherjee, tries to describe the history of cancer entwined with the history of cancer therapy. And it's a fabulous description of how drugs came along, how they were discovered, how no one knew how to use them until clinical practice, literally a decade and a half of clinical practice taught clinicians of how to use them. So many of the drugs, for example, that are used to cure kids with pediatric leukemia nowadays are the same drugs that were around in the 1950s, but people didn't know how to use them clinically.
Right.
And so the book really describes beautifully, the triumphs and the heartbreaks of all these different clinical trials, each one learning how to use the drugs a little bit better than the last one, and how long it took to come up with what we now call to be cures for pediatric leukemias. And in many ways, I sometimes tell people when they try and understand where is oncolytic virotherapy right now, oncolytic virotherapy reminds me of where drugs were in the 1950s. We have these powerful reagents, but we don't yet know how to use them clinically. In the lab, they look really awesome, but we don't know how to use them in people properly yet. But I think the day is coming when we will learn, and they will sort of join the tools that oncologists use to go after these diseases.
It's a great read to learn how we figured out how to use chemotherapy.
Absolutely. The stories are fantastic. I'd recommend that book to anyone. I really want to back up that recommendation because when I heard that TWiV, I just finished reading it myself, and I thought, man, what a great book.
This fellow is here at Columbia.
Yeah, and he writes beautifully.
He's a beautiful writer, yeah.
In fact, maybe a couple years from now, we'll get him to write a book on oncolytic virotherapy because he's great at describing how technologies come and how they have the potential to change diseases like that.
Well, he may be using it one day.
Let's get him on TWiV.
It'll be great, sure. Love to do that. I wonder if we could spend 10 minutes talking about the last topic here, destruction of smallpox virus.
Sure.
So give us a summary.
Or the lack thereof.
Or lack thereof, yeah. Tell us about that, Grant.
Well, it's quite a mouthful. You could easily devote a whole TWiV to the issue of smallpox.
We may at some point soon.
Yeah, give us the introduction and we'll...
okay. I'll set up the stage for you. I'm sure you know, and most of the readership or the listeners, I'm sure, also know that smallpox was wiped out as a human disease in the mid-1970s by the Worldwide Vaccination Program. And part of the reason is because it's another very host-specific pox virus. It only replicates very well in humans. It has no animal reservoir. So when the vaccine broke the chain of command from people to people, the human-to-human transmission, then it could be effectively eradicated from the biosphere. And so after smallpox was declared dead in 1980, the remaining stocks were rounded up and ended up being deposited in two repositories, one in CDC Atlanta, and the other originally was in Moscow, but then got moved to Novosibirsk near Koltsovo in the former Soviet Union, now Russia. So these are the two declared WHO repositories of live smallpox virus, live variola virus. And the use and the research that goes on with this live virus is under the auspices of a committee at the World Health Organization. And I actually sit on that committee, so I probably should disclose that. And So the WHO oversees all of the research projects that those two sites do with live smallpox. Well, after the virus had been rounded up and the disease had been essentially vanquished, at least from Homo sapiens, the debate began as to what should we do with the live virus? Should it be destroyed? Should we keep it and study it? And for a while, the scientific community kind of broke out into two camps, the pro-destructionists and the anti-destructionists. And there was a lot of debate. Should we kill the virus? Should we not kill it? Should we study it? Should we not study it? And the several resolutions have passed through what's called the World Health Assembly, which is the member states of the WHO, as to what should we do with the remaining stocks of the virus. And there's been a couple of resolutions that have passed over the years to, in essence, keep studying the virus until we have sufficient drugs and safe vaccines to be able to say that we've vanquished not only the virus, but the possibility of it ever emerging again, we'd be equipped to deal with it. And that debate is once again on the front burner because the WHA is going to vote again this year. I think it's in May, April or May, but I think it's May, on whether to request that the two member states holding the virus, the US and Russia, destroy the remaining stocks and the virus be declared vanquished forever. I don't know what the WHO is going to vote. Our committee at WHO was charged with putting together a document to summarize for all the member states what is the status of current knowledge about variola, what is the current status of the drugs and the vaccines that are currently available, and to put it all into a single document. And that is a public document now. You can get it on the WHO website, and it's been sent to all the member states, and they're all conferring amongst themselves, as I presume, as to how they're going to vote when it comes up to the WHO assembly.
Can you vote yes on destroying the virus, but no on declaring it vanquished forever?
Well, you probably could, because the actual vote is simply.
Because I'm thinking that might be a wise choice.
Well, one of the things, that is, I think, relevant to this issue. Last year, I wrote a commentary in PLOS Pathogens. And I should mention I'm the deputy editor-in-chief of PLOS Pathogens, so this was one of my prerogatives, is to be able to write a commentary on something that I thought people should think about. But the purpose of the commentary...
Grant, you get a prize for the record number of disclosures on
Yes.
Yes, sorry. But I think I
great.
should tell listeners these different disclosures, because in some of them, there's a reason why I have my opinions about it. But one of the purposes of that commentary was to point out to people that even if the virus is destroyed right now, the advances of synthetic biology are such that if someone wanted to create smallpox virus from scratch, it's now technically possible to do it simply using information that's there in the scientific literature. So
but it would be
that's
an
a complication.
step they would have to do.
Sorry, I didn't hear that.
It would be an additional step. I mean, one of the arguments for getting rid of the stocks is it would prevent somebody from simply swiping them from a freezer and wreaking havoc. You'd now have to go to the effort of synthesizing the virus.
Correct. And in today's world, that would be like it would be doable, but it'd be technically, not a simple operation. But, there are many labs in the world that could do it right now. But 100 years from now, who knows?
Right.
You might be able to just dial up on your cell phone, for all I know.
There is a editorial in Vaccine, why not destroy the remaining stocks? And their point is, they say the simple fact is that smallpox can now be reconstituted from sequences. But in fact, that hasn't been done. It's theoretically possible.
Correct.
But it could be tough, as you say, right?
Yeah, there's still probably some technologic challenges that would have to be worked out. But there's no reason to think a lab couldn't do it right now. It would just simply take the time and the effort and the money it would take to do it at the moment. And the point about synthetic biology is we don't know how fast those technologies are going to evolve in the future. But given the precedence of all other technologies that are sort of like that, our best guess is it's going to be easier, faster, and cheaper in the future than it is now.
So, Grant, there was a rumor a long time ago that there are some missing vials from the vault, so to speak, particularly from the Russian side of things. I'm obviously a good friend of Steve Morrison. We used to teach a course together called Emerging Infections, and one of the hints was that even if you destroyed everything that's known, there still might be some unknown vials hanging out someplace that we're not aware of. That during the turmoil of converting the USSR to the country of Russia, that these vials may have just walked off someplace. Do you have any information on that?
I have no information other than it's the fears of many people and certain governments, but there's no hard data.
Right.
But it's one of those things you can never prove that they don't exist.
Isn't that the formation of the basis for the hesitancy of getting rid of the ones that we have? Because once we do, it gives a big advantage to the missing vial hypothesis.
you could. It depends how paranoid you really are.
Oh, exactly.
Well, on the other hand, I think it's worth pointing out, if people forgot from 2001, there was a little incident with the Postal Service.
Yes.
With weaponized anthrax being sent around, and there were some criticisms about the investigation and yada yada yada, but I don't think there's any serious doubt it was an inside job. Mm-hmm. So if you have stocks of smallpox, you can't ultimately protect against that. So no matter how secure your stocks of smallpox are, and even if there aren't these unaccounted stocks floating around, that's a danger.
Yeah, I mean, the stocks that exist now are a danger. They could be stolen in the future.
Yeah.
The whole thing is a risk versus benefit ratio. It depends where you are or in your opinion.
So, Grant, the vote that is going to be taken, is that a binding resolution? So the countries that are told, do they have to destroy the stocks, or is it a choice?
Then you're getting into the political arena. It isn't so much science.
Okay.
Yeah, we're out of the realm of science.
What would happen is that WHO were to send its recommendation or mandate or whatever to the two countries, how they will respond to that.
Right.
It's in an arena that has little to do with science.
Here.
Interesting. And then maybe one country would do it and not the other, or vice versa, or both would or both wouldn't.
Right. So usually these sorts of things, they have some sort of mutual assurance protocol where you visit the other site and all that, but still.
Come on, it's like destroying nuclear warheads.
Exactly.
I think that editorial in Vaccine even had something about that almost sounds like nuclear arms limitation.
Yeah, exactly.
I'll get rid of 2 vials if you get rid of 2 vials, right?
Yeah,
well, and in fact,
all you need is one particle.
There's a similar argument here that if the U.S. and Russia are holding on to this stuff, then it gets a little dicey when we try to tell other nations, oh no, you shouldn't have this.
Well, sure.
Mm-hmm.
I think, Grant, maybe you can comment on this, but I think one thing that people don't fully appreciate is that we're not just talking about one strain of virus. We're talking about a repository of hundreds, at least, strains collected over time and globally. So there's here, and not all of them have been studied, not all of them have been sequenced, and so there is an incredibly rich repository of information about virus virulence, pathogenesis, evolution, etc., that has not been tapped.
Yes, and in fact, it's very hard to tap some of those things right now with our current technologies. And one of the big unknown issues is, what will the technologies be like in the future? Might some of those things be tappable in the future? It's just one of those very interesting questions that melds science with politics and philosophy and ethics.
Sure. What's the closest pox virus to smallpox as we talk now?
It depends
Camelpox.
how you define it.
Yeah.
The
Genome.
closest disease is monkeypox.
Monkey. No, that's what I thought.
But it's not the closest genetically.
No, I was thinking
genet-
of one making the jump between where it is now and back into humans. You know, like SIV into HIV, that sort of thing.
The closest genetic sister virus, if you like, is probably camelpox or another virus that's sometimes called gerbil pox. They're the closest genetically if you were to imagine somehow getting a virus to become more smallpox-like.
Right.
But the only one that really causes a disease in humans that looks like smallpox is monkeypox.
Monkey, right.
I didn't realize it was a whole collection. I thought it was just one virus, maybe 100 vials or something. And
No.
Wow.
We're talking
No,
about,
there
we're
are
talking about
hundreds
a
of
huge
samples.
repository.
Wow.
We're talking about virulent strains with varying virulence. We're talking about variola minor, variola major, things from all over the world. It's an incredible repository.
So it's harder to keep
That
track of
makes-
than we thought.
But that makes it different. It's really
Absolutely.
it's really a resource, and I think most people don't know that.
Yeah.
So what
That's
I'm
a good point. That was not certainly. I don't think that was expressed in the virology editorial.
No, it all sounds like just smallpox. That's it.
yeah,
Right.
Yep.
that's a good point.
Yeah, and I think in
many
the public
ways,
mind, it's pictured
those
as
samples.
one bottle.
Yeah.
What were you going to say, Grant?
Sorry.
I was just going to say that in many ways, those samples were taken from patients all over the world at various times, and they're like the last snapshot we have of that disease and that virus on the planet.
By the way, they reconstituted the influenza of 1918 by simply digging up a frozen body and sequencing a lot of different
Not so
strains.
simple. Not so
No, it wasn't simple, but they did it. Are there any possibilities of ancient samples of smallpox around the world, like, from mummies and stuff like this, that might still be viable?
People have worried about it in bodies that are in the tundra.
Indeed.
In other words, that have been frozen solid for like decades or possibly even centuries. People have gone to look for live virus in such bodies, have not been able to find it or resuscitate it so far. But that doesn't mean it couldn't exist.
Exactly.
I think what I will do is put up a poll on the website and say, go
good
listen
idea.
to TWiV
Yeah, great idea.
124
Yeah.
Yay or nay?
between X and X minutes.
That's right.
And then vote, because I think most people don't realize it's a repository.
We need some sort of a link to your document that you wrote for the World
Yeah.
Health Organization too, Grant.
Yeah. Okay. Yep. Can I dig it out?
Let's end this with the last sentence of Grant's Ploss opinion. The debate may also prove to be instructive when the next human microbial pathogen lands on death row awaiting our collective verdict.
Ooh,
It could be polio virus.
it could.
I have been threatened with eradication of my stocks for years now. And now I'm realizing may not ever happen. Because even if we do eradicate it,
It won't eradicate
maybe
it.
not destroyed. I don't know. Grant, what were you going to say?
But this decision that is happening right now with smallpox in many ways will be the template of the argument used with polio.
Sure.
You bet.
Yeah.
Yeah.
You bet.
But, I mean, as we've said, it may not be destroyed, even
Gentlemen,
pending any vote. So.
Doesn't anyone here sense the irony of all this, though? Here we are dealing with an incredible pathogen that we are tempted to get rid of. And yet, by our very activities on Earth, we have extincted countless numbers of other life forms. Isn't that, by contrast, something to think about?
Yeah, but they
Yes.
don't kill us, though, Dickson.
You don't know.
What are the moral implications of viricide?
Yeah, exactly.
I'm not morally worried. I'm just worried about this repository.
Right. The loss of data is what's worrying.
Yeah.
I mean, but to focus our attentions on such a small issue when the larger issue is how do we prevent the loss of biodiversity? Just when you compare it to the larger picture, it pales by comparison. And yet this is This Week in Virology, so we got to talk
Yes.
about viruses.
Sure.
Sorry
Okay.
about that.
No, it's good coda there, Dickson. I think we have time for a couple of emails, so let's start with the one from Norm. First, I love your podcasts. You guys are constantly grousing about the lack of research funding. I was curious. NIH funding has risen every year since 2000, from 17 to $31 billion, supporting 325,000 researchers. NSF funding has risen every year since 2000, from 2.5 to 5.5 billion. I assume money is also available from corporations, charities, foundations, state governments, foreign governments, Department of Defense, CDC, FDA, universities, etc. Looks to me like we are spending lots of money on research. I wonder, is it possible that we are not spending money on the right things because politics and bureaucracy push the money to the wrong things?
Oh, dear.
My local government spends lots of money, but strangely never seems to be able to fix potholes.
That's right.
Some people say we need to give the government more money so they can fix the potholes, but when they get more money, the potholes stay unfixed.
Right.
Strange that. I am suspicious that you could double the funding of NIH and research you advocate would still not get funded, and you would still be complaining about too little money for research. As a last thought, it seems to me that the cost of research should be going down. After all, much of modern research equipment and services are computer-based, and these should get cheaper and more powerful every year. Am I missing something?
You're missing several things, Norm. I'll start off with the economic issue, which is that anytime you talk about money over time, you need to use an inflation calculator.
Here.
And the Bureau of Labor Statistics has one online. You can plug in the numbers and find that $17 billion in 2000 is $21.74 billion in 2011 dollars. So the NIH funding has not risen quite as much as you say. It went actually from 21.74 billion real dollars to 31 billion. Now that is still an increase.
Oh, yeah.
But that gets to the other issue here, which is we're not spending money on research. We are investing money in research. And I've seen a couple of economic analyses of some of the things that have come out of biomedical research. And if you count the economic benefits that we get from, say, the polio vaccine, and these are accruing benefits that occur every single year because of all of the kids who are not paralyzed and all of the people who have not died, and all of the additional economic activity and the all of the medical care and iron lungs and what have you that we have not paid for because of this vaccine. And if you tally that up, I believe the calculation was that it alone would bankroll the NIH indefinitely. And that's just one discovery. So if you like your cell phone and you like your computer and you like the internet and you like living to the age of 80 instead of 40, you are a tremendous beneficiary of this pittance of our gross domestic product that has been spent on research.
Yep.
This is not even, we're talking about rounding errors in something like the defense budget. So the amount is actually still extremely small for the benefit that we're getting. And what we're talking about here, when this does come up, which I don't think is quite constantly, is that more should be invested in research because of the benefits that it has empirically returned over the years.
There is an issue here. He says, is it possible we're not spending the money on the right things because policy and bureaucracy push money to the wrong things? There have been difficulties at times with the legislative process earmarking money out of the NIH budget for this and that has without allocating any more, and that has the effect of basically politics manipulating the NIH budget, and that's not good. Okay.
Right.
I'd also add that in my own experience that the numbers of times that you have to replicate the wheel in order to do the same kind of research in different laboratories argues in favor of some kind of a centralization for certain things, which has trended towards that in terms of sequencing of DNA samples and proteomics. And for instance, you can actually send your samples out now to have them done rather than having to have the equipment sitting in your laboratory. But for a while there, big science requires bigger funds and more people, and the whole thing just snowballs. And if you want I'm sorry. No, I'm just going to say, if you want to do real science, it's a lot more expensive today than it was 10 years ago.
Right. I don't think the cost goes down.
Right, and the issue here is, the costs of the $2,000 computer that you're using in your lab has the performance of it has increased. The cost has not gone up, so that's great. But the $300,000 mass spectrometer that you're connecting to that computer has gotten progressively more complicated and more expensive. And the result of that is you can get more data out of it, but then you are also expected to get more data because that's where the science goes. I mean, if you look at sequencing, which Dick just brought out, that's now a commodity service. You send off something to be sequenced. 30 years ago, sequencing a kilobase would get you a paper in Nature.
Correct.
Today,
and a job at a university.
Yeah, exactly. Today, sequencing a genome might be enough if it's a big enough genome. It's practically to that point. So more is expected as more can be done, and that's why we end up because we keep discovering more and more complexity that we need to figure out.
The biggest lament, I think, of all of us ever has been the proportion of those who applied for research funds versus the number of people who actually got them. And that's the biggest grouse that I have. I mean, you have lots of ingenious people out there with wonderful education, and they apply and they put their heart and soul into these applications, and they end up on the floor in a box that you've just sat down at the NIH and reviewed, and you've got to do 100 or 200 of these before the study section finishes. And how many did you fund, Vince? Last time you went down there?
I don't know. We don't fund, but it's not even 100.
Recommended for funding?
It's not even 100 anymore, and maybe we review 70, and I'll bet 10 get paid.
10.
That would be my guess. Tops.
Right, and it's not just that people have poured their heart and soul into these proposals. It's that some of them are brilliant and might have given you breakthroughs we could use.
There's really good research that's not getting funded.
That's the biggest tragedy. I mean,
so he's got a sentence in here too that says, "I'm suspicious that you could double NIH funding." Still not get funded and still be complaining. There was a period of time. When was it, guys? In the 2000s.
In the 2000s, actually. It's. I think he chose the 2007. I think.
When the NIH, there was actually a government mandate to double the NIH budget over a period of 5 years.
Yep.
And nobody was complaining, and a lot of people got funded.
That's true.
Yeah, and in fact, part of the problem now is I think that was the 90s. That was the late 90s, wasn't it? And it ended in the early 2000s. And one of the big problems was that they didn't just say, okay, we'll double the NIH funding and then we'll keep up with the cost of living and inflation and have a steady increase and here's what's going to be going on. Yep. It was we'll double the NIH funding and then bam, we're going to flatline it.
Yeah,
and
that's like the
hard landing. That
the stimulus funding. That's right. The stimulus money was the same.
Yeah. I would add that despite most years it is flat, and when it does go up, it covers inflation, and labs use more money, so fewer people end up being able to go into science.
Right. The inflation calculation I did was based, just on general inflation. The actual inflation in the cost of doing research is. I would expect considerably greater than that.
And there are proposals being considered in the legislature right now for the next year to actually cut the budget.
Yeah.
Yeah, that's right.
And I've never gotten money from most of these sources, mainly NIH and NSF. No, I never got an NSF grant, although many people I know get them. So out of that long list, in fact, what's available to us is mainly NIH and NSF.
NIH is really the deep pockets.
It's the big one, yeah.
And corporate grants usually want you to do what they want you to do.
Sure, it's very focused. All right.
We'll stop our grousing.
I think we should do just one more, and that is from Lynn Enquist, our colleague at Princeton, whose paper we discussed a few episodes ago. I enjoyed hearing you discuss our recent paper on some basic virology using colorful herpes viruses. I have some answers to questions you raised about the phenomenon of limited viral genome expression and replication. So just to remind everyone, they had made colored herpes viruses expressing 3 different fluorescent proteins of 3 different colors, and in co-infection experiments made the interesting observation that no more than 6 or 7 genomes seem to replicate in a given cell, no matter how many you put in. First, the FACS sorting experiment says that the viral genomes that are being expressed in the cell are the same ones that are replicated and packaged. Expression, replication, packaging are all linked. You don't have a pool of genomes all expressing things and a subset get replicated, or a random pool of genomes replicating and expressing with a subset being packaged. Rather, a few genomes are chosen early to be transcribed, replicated, and packaged into virions that go on to infect the next cell. So that's very interesting to me. I wonder how universal that is, because it is I'm surprised, actually.
And how would the selection occur? And
yes.
How does the cell know which ones to permit and which ones not to?
Or what does the virus do?
Or the other way.
To narrow the field.
First one in, last one out? First one in, first one out?
Maybe. I don't know. It's a good question. Second, Oren has shown that the effect is not due to the promoter used. He did a beautiful set of experiments where the pseudorabies virus VP26 capsid protein was fused to each of 3 different fluorescent proteins, and these hybrid genes replace the normal VP26 gene. These hybrid genes are transcribed from the VP26 promoter, which is a pseudorabies virus promoter, expressed only after DNA begins to replicate. Cells infected with mixtures of these viruses reproduce the same phenomenon as we reported in the paper. They also provide more data since these fusion proteins assemble into capsids in the nucleus. The dots of colors in each nucleus are all the same color, verifying again that only a few genomes are chosen to be expressed and replicated. Amazing, I think, and very colorful, too, for Rich's benefit.
Neat.
Oren has infected primary neurons, primary fibroblasts, and other cell lines with the colorful herpes viruses we have on hand. All cells show the same restriction of color as we reported. So I'm interested to know how many other viruses do this as well. And we, in fact, are starting to do this with one of the viruses we work with.
Nice.
All right.
I think we lost Grant again.
Did we?
Oh, dear.
You can't hear me?
Sure we
Oh,
did.
you're there. Okay.
He's just
Sorry.
he's soaking it up.
Yep.
Just soaking it up. Okay.
So let's move on to picks of the week.
Oh, good.
Grant, do you have a pick for us?
Sure. Actually, when you first asked me to join, I was going to pick that book on cancer,
Right.
The Emperor of All Maladies. But since you've done that, you've preempted me. I thought I'd go to the opposite end of the spectrum. That book is really, illuminating and thought-provoking and moving, actually. So I'd pick something lighter and maybe funnier. And there's a book I read a few years ago that I still love. It's called A Short History of Nearly Everything by Bill Bryson. And it's a great read. It's a few years old now, but I think it's in paperback. I read it originally as a hardcover. But Bill Bryson is he's not a scientist. He's a very funny writer. He likes to travel around the world and report on his experiences. And he has a really funny book about Australia called In a Sunburnt Land. But somehow he got interested in science, and I don't know how it happened, but he decided to write a book about science. And that's this book. And he covers a whole swath of areas of science, everything from physics to biology. And he has a particular predilection about the quirkiness of human beings. And he loves to sort of talk not only about what scientists have done, but how, in many cases, what oddballs they were while they were doing it. And it's a wonderful mixture of humor, but at the same time teaching you about how the scientific method actually works. It's a great book if you have any relatives or friends who aren't scientists who sort of look curiously at what it is you actually do for a living. You can give it to them, and nearly anyone can enjoy the book, but it's very sly at how it teaches science along the way.
Sounds good.
Yeah, we actually had a quote here a couple of weeks ago by when we were talking about whether to trust scientists or not, and the answer is trust science, don't trust scientists.
Yes.
Yeah.
Right. It's good. It's available on Kindle, so...
Yep.
Bryson also wrote another book that I've read called The Life and Times of the Thunderbolt Kid.
Yes, which is a great read as well.
And he also wrote A Walk in the Woods.
Yeah.
Yes, that was
Look
his
at
first book.
He's actually quite a prolific writer. There's a bunch of stuff.
We are among literati here.
Thank you, Grant. Dickson.
Oh, my pleasure.
Dickson, what do you
I
think?
have something here, Vince, because I've just moved in next to you and I've unpacked all my books again for the 400th time. I have a reprint of Edward Jenner's original Inquiry into the Causes and Effects of the Variole Vaccine,
Excellent.
Awesome.
published in 1708.
I've actually read this.
And this is
And
a
it's
fantastic. It's illustrated too, and it's got all these beautiful diagrams of what a pox looks like and what a cowpox versus a smallpox vaccine looks
That's
like.
terrific.
And it's a
I've
wonderful,
often wondered
wonderful
whether
book.
he himself did those illustrations.
I don't know, but they're gorgeous, and
beautiful.
I've admired this, and maybe that's where, I'm a viral appreciator rather than a virologist, so I come to you with my arms open, and this is a gift.
Well, this is interesting. It's actually available on Amazon.
Yeah, I just looked it up and put the link in there.
Cool.
There's an illustrated edition.
Yeah,
Well,
I've got the illustrated
Dickson's
editions.
is dog-eared and
It's well read.
it looks very old, even though it's not.
It almost looks like
It's
the
a
original.
fascinating read.
Yeah, it is.
He goes through the whole thing. It's really terrific. It's a great pick.
Totally agree. Thank you.
It's really appropriate for this episode, too, Dickson.
That was an emergency pick, but I'm glad I found it.
Emergency pick. Dickson, you have a pick? No.
Yeah, that's what he said to me to begin with, and I remember I just gave him this book.
Rich, what do you have for us?
So I'm passing on a pick that was sent to me by a student, Megan, who I've encountered in a number of circumstances. She's my beekeeper friend.
Oh.
And most recently was in our virology class. She sent me a thing that's on a subdomain of the TED site that is a commercial that was for the Topsy Foundation, which is an AIDS advocacy foundation. And it's beautiful. It's, I have to just, I'm at risk of stealing the effectiveness of this, but I have to do this. It's a time-lapse photograph of a woman undergoing antiretroviral therapy for AIDS. Okay? They talk about, they basically say this is what AIDS can do, and show you a time-lapse of pictures going from day 90 backwards to day 1. Okay, you're seeing a person getting sick as they go from looking healthy to being wasted. But then you get back to day 1 and you realize that on day 1 they started the antiretroviral therapy.
Wow.
And it's really stunning. It's just a couple of minutes long, worth a look.
Neat.
And the Topsy Foundation is interesting too. They're worth looking at.
Wow. Nice. 1 minute and 35 seconds, yeah. Shorter than TWiV.
Shorter than
A lot.
A lot shorter.
Shorter than TWiV.
Alan Dove.
My pick of the week is there's a Times story about it today, and I've also got the link here for the report that stimulated the story. The draft report on WHO's response to the H1N1 pandemic just went online yesterday. And this is the WHO convened a panel, an independent panel, to essentially audit their response to the H1N1 pandemic. And as people are probably aware, the organization received some criticism from during the pandemic. So this panel looked into all of this, and it is summarizing it. And this is a draft report. It now has to go through public comment period and all of this. But the Times article that summarizes it gives a pretty good flavor for what's going on. I will warn people, if you want to read the whole report, I haven't gotten all the way through it. It's
That's right.
It is definitely a WHO report. No doubt about that. But when you look through the careful couching and diplomatic language of it, and if you manage to keep your eyes open, it kind of tears the organization a new one about some issues. But at the same time, they investigate and dismiss a couple of the more extreme accusations that people were making at the time. So it looks like it was a fairly good analysis of what went wrong, and I guess the diplomatic term is that they have a list of lessons that they have hopefully learned from this.
Well, it's interesting in a way, or good in a way, that we had a pandemic that turned out to be not too virulent a virus.
Yes.
As a sort of a test case, and they could look at it and say, how did this work, and how can we improve it?
Right.
Yeah.
I like here Harvey Feinberg, who was the head of this agency, said the agency's needlessly complex definition of a pandemic with 6 levels of alert.
Yep.
Yeah, this is very interesting.
Somewhat confusing.
I don't know if anything will change, though, because at WHO is just impossible. It's too big.
Talk about politics.
That's the problem, but that's very cool.
I think that was the intent of this draft, is to really kind of shake things up a little bit.
Yeah, the Times is a good summary because, as Alan says, the original report is quite dense.
Yeah.
Okay, my pick was suggested by Al, who wrote, "Dear Twivers, forgive me if this has already been mentioned on TWiV. I'm catching up from the beginning, and I am on episode 30.
He is the way to go.
Great, but wanted to share this gallery of science visualizations at Wired, which includes a lot of microbiology images, starting with an impressive image of an HIV particle. Enjoy and keep up the great work." So this is a Wired summary of the science contest, which was Science and Engineering Visualization Challenge of 2010. Beautiful images, and the first one is HIV,
Right.
which is gorgeous. Just shows you how sparse the glycoproteins are in the viral envelope.
Well, Al, spoiler alert, everything turns out okay with H1N1.
Ah.
Right.
Yeah, 30
So
is
it
a long
was
time
much
ago.
ado
That's
about
right.
nothing after all.
Much ado about nothing. Well, Dickson, you said it wasn't going to be a pandemic anyway.
Well, but my definition was number 7 that they didn't even
They never
include in
got to
there.
that. Okay. And that will do it for TWiV. Don't forget to go to facebook.com/thisweekinvirology, our fan page, and check it out if you want to listen. iTunes, Zune Marketplace, or the Microbe World app for streaming it to your iPhone or Android device. Or you can go to twiv.tv and play or download our episodes and read our show notes. We always love to get questions and comments. Send them to twiv at twiv.tv, or you can call up our phone line, 908-312-0760. Grant McFadden, good to have you back again.
My pleasure. It's great talking viruses with you guys.
Excellent.
Terrific.
You're natural. And we'll have you back again when we destroy all the smallpox stocks, or maybe not.
And then we go after polio.
Yes, and then I can complain. Dickson Despommier, thank you.
Pleasure to be back.
Verticalfarm.com. How is the vertical farm?
It's growing up.
All right.
Actually, they've got plans for one in Milwaukee that's, I won't say that it's gotten its full funding yet, but they've gotten lots of interest from venture capitalists with a good heart. So I think they'll get it done.
Beautiful. Glad to hear it.
Yeah.
Alan Dove is at alandove.com. Thank you.
Always a pleasure.
Wonderful. And Rich Condit is at the University of Florida, which is also where Grant is. Thank you, Rich.
You're quite welcome. Always a good time.
Happens to be the home of the Gators.
That's right.
There you go.
Grant, you're not a football guy, are you?
Well, it's hard not to notice football in baseball.
That's for sure.
How did Florida do for the ACC tournament? Or the no, not the,
sudden silence.
It was I watched
The basketball tournament?
Yeah, I started to watch, but then I turned it off and got interested in something like volcanoes or something.
I don't pay any attention.
It's football. So you don't go to the games with Rich and Moyer, right?
Not yet, but the day is young.
You might. All right. I am Vincent Racaniello, and I'm at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We will be back next week. Another TWiV is viral.